this is Lynn, and we are going to cover the muscle physiology in this module. The learning objectives are as follows. Describe the organization of muscle tissue and contrast the structure of skeletal, cardiac, and smooth muscle. Explain how muscle striations are created and how the regions change during muscle contraction. Connect all the molecular structures to the contraction phase that they influence. Describe the sarcoplasmic reticulum and its overall functionality. Distinguish the main steps defining muscle contraction to relaxation cycle. Explain the molecular basis of muscle contraction and cross-bridge cycling. Describe the mechanisms of excitation-contraction coupling with respect to the ryanodine receptor and the activation by the dihydropyridine receptor, diagramming the roles of calcium plays as a secondary messenger. Differentiate the process and pathways involved in the stimulation, excitation, and relaxation of each muscle type, describing the function of different components found in each muscle tissue. Alright folks, let's get started. So the characteristics of skeletal muscle tissue. Skeletal muscle has the ability to be excitable, respond to the nervous system, conductible, meaning it can pick up a change in electrical charge, contractile, it has contractile proteins to allow the contraction to occur, it's extensible, it can lengthen to a certain degree, and there's also elasticity bound inside. The microscopic anatomy of a muscle fiber is as follows. It creates a striated appearance because of its appearance under the polarized light, giving this dark and light banding pattern. It is fused with numerous cells together. That's why you see this multi-nucleated system. Numerous nucleuses are found in a muscle fiber. The cytoplasm of the muscle fiber is called the sarcoplasm, and they're filled with specialized contractile proteins that give it this ability to slide on top of each other. The sarcolemma represents the plasma membrane. The plasma membrane also has these invaginations, these deep tubes called the T-tubules, transverse tubules, which allow signals to propagate to the deeper parts of a fiber. Along the length of the sarcolemma, as well as the T-tubules, it's filled with numerous sodium and potassium voltage-gated channels so that this signal can propagate down. 80% of the muscle fiber's volume is composed of these long cylindrical structures called the myofibrils. And within the myofibrils, we have these single protein filaments called myofilaments. The sarcolemma is the plasma membrane of the skeletal muscle fiber. The sarcoplasmic reticulum is a uniquely modified internal membrane complex that is analogous to the smooth ER of a general cell. It encases each of these myofibrils like a sleeve of membrane netting. They also have these blind sacs at the end of each sarcoplasmic reticulum termed the terminal cisternae. When you have two terminal cisternae lying adjunct to each other, including the T-tubule, you form a triad, which means it's one tube, second tube, and three tube, and this is called a triad. Within the sarcoplasmic reticulum, there's a high amount of calcium ions, which is very important for contraction purposes. There's also a protein called calsequestrin, which is able to reuptake and store 40% more calcium when contraction is not happening. The T-tubules are in intimate contact with the terminal cisternae of the sarcoplasmic reticulum. It contains these very important voltage-sensitive calcium channels known as the dihydropyridine DHP receptor. There's going to be a charged amino acid residue when the DHP is activated that causes a change in its shape. On the right-hand side, you can see that as an action potential travels along the membrane, this DHP receptor, which picks up the change in voltage, will then activate to the internal compartment that is attached to the door, which is known as the ryanodine receptor, which will then open up to allow the sarcoplasmic reticulum to influx the calcium into the cytosol. The calcium release channels are controlled by that door called the ryanodine receptor. These channels can be open for a few milliseconds, and they quickly will close down when contraction is over. They generate this excitatory pulse of calcium ions. So calcium is a very important regulatory component enabling muscle to contract. Over here is a picture showing you the calcium pumps after contraction has taken place. Calcium requires energy to be restored back in to the sarcoplasmic reticulum and then held by the calsequestrant proteins. Let's take a look at the myofilaments. Myofilaments are the contractile proteins that are bundled from end to end and extend the entire length of the myofibril. There are two types. We have the thick filament and we also have the thin filament. The thick filament is composed of about 200 to 500 protein molecules of structure, and they consist of this actin binding site at the tip of the head, and we also have an ATP binding site at the body, as well as light chains and heavy chains. It almost looks kind of like a golf club. The head serves as the ability where the catalytic ATPase activity occurs and ATP attaches to and gets split into ADP and pyrophosphate. On the other side, a thin filament is composed of about 300 to 400 smaller globular G-actin proteins, as well as this fibrostrand called the F-actin. More importantly is that it has the myosin binding site where the myosin head will attach to. It also includes two proteins called tropomyosin and troponin complex. You can see on the right-hand side that the two heavy chains are coiled together in the myosin head, and then we have the four light chains. And then in the center, we have a hinge region which permits the sliding filament and tugging so that the myosin and actin can overlap and allow the sliding to occur in muscle contraction. Organization of a sarcomere. Sarcomere is the smallest contractile unit in a muscle. Sarcomeres are myofilaments within the myofibrils arranged in these repeating cylindrical, roughly about 2 micrometers of distance, but this is the smallest contractile unit inside a muscle. From each end to each end, the border is called the Z line or Z disc. This marks from one end frame to one end frame that is perpendicular to the myofilaments and the thin filaments. You can notice that there's this darker region where we have the A band. This is overlapping of the thick and thin filaments, and it contains two other compartments, the H zone as well as the myomesin M line, another structural protein that anchors the filaments together. The I band is bisected, divided by the Z disc, and contains only the thin filaments. And that is why you can see in the picture that's only the actin filament as well as the titin shown here in this cartoon image. The thick filaments are shown here like golf club heads. The overlapping part is the A band, and then the lighter part that doesn't have the overlap is the I band. The overlapping of myofilaments form these lighter and darker regions, and thus appearing striated due to the size and density differences. Striated muscle indicates that it has a potential to contract. There are also other structural proteins involved, like titin. That's this big coil protein, largest single protein in the human body. Well, it looks like a spring. It's used to stabilize and align the thick filaments. It also provides majority of the elasticity. Another protein that you don't see on the screen is called dystrophin. And dystrophin is a protein complex that also sits adjacent to the sarcolemma and maintains the attachment of the inner filaments to the external proteins of the cell membrane. When this protein is damaged, the muscle begins to waste away as a result in a condition called Duchenne's muscular dystrophy. Excitation-contraction coupling. Motor units are composed of a single motor neuron in the muscle fiber it controls. The motor unit has an inverse relationship with the degree of control, such that the smaller the motor unit, the greater the degree of control. For example, in your eye muscles, there's a lot more fine-tuning and quick action versus the muscles inside your quadriceps or your hamstrings. Motor unit muscle fibers are dispersed throughout the muscle rather than clustered and produce a strong contraction in a localized area and a weak contraction over a wider area. So one single muscle may have many motor units. Alpha motor neurons are what innervate the muscles from the CNS. Some of the small motor units are composed of about 5 or less muscle fibers, and the larger ones could have up to 1,000. The axons of these motor neurons are generally myelinated to allow quick response. On the right-hand side, you can see an electron micrograph picture of the muscle fibers. You can see the NMJ as well. They're not physically touching, but they're very close to the muscle fiber with the axon motor. And then on the right-hand side, you can see the spinal cord allowing this intervention to the muscle, where certain fibers get innervation from certain branches of that motor neuron. The NMJ is the neuromuscular junction, a small region where the nerve is able to communicate to the muscle, which we'll talk about in the next slide. Neuromuscular junction. So each muscle fiber has one NMJ, or specific location in which that fiber is innervated by a motor neuron. The NMJ has three general parts: a synaptic knob, the motor end plate, and the synaptic cleft. The expanded tip of an axon is also known as that synaptic knob, which is enlarged and flattened and covers a good amount of area of the sarcolemma. Let's take a look at the steps involved in activating that signal to propagate through the muscle fiber. Keep in mind that all NMJs are excitatory in nature. The first physiological event of skeletal muscle contraction begins by that somatic motor neuron releasing the right neurotransmitter. In order for it to do that, the calcium begins to influx into that synaptic bulb at the tip end. As you can see over here, the calcium goes in via these voltage-gated channels, and they're trapped very closely to these dense bar linear proteins at the tip of the axon butins. The calcium eventually will bind into the synaptotagmin complex, as we talked about before in the vesicular release. That will then allow the acetylcholine neurotransmitter to begin to serve and dock onto that terminal membrane. Roughly about 125 vesicles are docked. The acetylcholine neurotransmitter is released via exocytosis, and it begins to flood into that local area. It will then excite the muscle fiber and continue on until the acetylcholine esterase begins to break that down. Acetylcholine will then bind to these specifically chemically gated receptor channels at the motor end plate. That will then begin to allow the sodium ions to start to go in. And because you have a greater amount of sodium influx than potassium efflux, there is a net positive charge that begins to develop in the cell membrane right here. In this visual, you can see that in the depth region on the muscular membrane, you have these synaptic gutters where they're entrenched with numerous voltage-gated sodium channels, as well as the acetylcholine receptors. So as a neurotransmitter gets released, a lot of it gets trapped into these gutters where the signal can propagate. As a result, the motor end plate is created, also known as the end plate potential. This ranges from about -50 to -75 millivolts. And this end plate potential, EPP, is the minimal voltage or threshold needed to trigger more voltage-gated channels in the sarcolemma to initiate an action potential. The moment you can reach that threshold of about -50 to -75 millivolts, the muscle fiber will go generate an action potential that can propagate onwards. On the right-hand side, you can see the acetylcholine receptors with the negative charges that repel against positive ions from entering in. So the moment acetylcholine binds, it opens up and allows sodium to rush into the cell. Troponin and tropomyosin are regulatory proteins associated with the actin and make up the entire troponin-tropomyosin complex. Tropomyosin is a short, thin, twisted filament protein that covers the actin strands and the myosin binding sites on the non-contractile muscle. Troponin is composed of three subunits. It has the I inhibitory, which binds onto the actin, so no interaction can occur. The troponin T is a tropomyosin binding activity between the two parts, and the troponin C is a segment of the protein that binds and interacts with 4 calcium ions. Troponin and tropomyosin both together cooperatively block the cross-bridge sites from forming. On the right-hand side, you can see that when you have low amounts of calcium, the muscle is in a relaxed state. This is because the troponin is covering over the binding sites, and the troponin has no calcium available bound to it. When calcium level increases, the calcium ion will dock onto the actin. That will then open up and twist the tropomyosin complex in a conformational change, exposing those binding sites so that now the myosin head can interact with the actin filament.